34 Israeli aircraft are on the ground simultaneously. In the next 36 hours, Operation Solomon will lift 14,325 Ethiopian Jews, the Beta Israel, the Beit Yisrael, the House of Israel, out of a country in the middle of a civil war. One El Al Boeing 747 will carry 1,122 passengers on a single flight. That world record still stands. But inside every one of those 14,000 bodies traveling at 37,000 feet over the Red Sea is a chromosomal anomaly that genomic science will spend the next 30 years attempting to explain, and that a court will spend the next decade attempting to use. In 2010, the largest genome-wide study of Jewish populations ever conducted compared 14 Jewish diaspora communities against 69 non-Jewish populations worldwide: Ashkenazi Jews, Sephardic Jews, Mizrahi Jews, Middle Eastern Jews, every group clustered tightly together on the genetic map, overlapping with Druze and Cypriot populations, tracing cleanly to the ancient Levant. The Beta Israel did not appear in that cluster at all. They sat in a different region of the genetic map entirely, close to non-Jewish Amhara and Tigrayan Ethiopians, close to the people they had lived beside for 2,000 years, not with the Levant, with their neighbors. Before you decide what that means, and you've already started, there is a second finding from the same study. It changes everything about the first one. The same 2010 study that placed the Beta Israel outside the Jewish genetic cluster also documented something in their Y chromosomes, their paternal line, father to son, unchanged across every generation. It pointed west, toward the ancient Levant. The genome-wide picture, driven primarily by the maternal line, pointed south and east into Africa. Two inheritance systems, one community, two completely different continents, both signals confirmed, neither one a measurement error. Early Y chromosome studies found roughly 41% of Beta Israel men belonging to haplogroup A, the oldest documented male lineage on Earth, tracing back over 140,000 years into the East African Rift Valley. But a documented minority carried haplogroup J, the West Asian signature found at its highest densities in Levantine and Arabian populations. These were not outliers contaminating a clean dataset. These were the two genetic inheritances of one people, living in the same highland villages, around the same lake, practicing the same pre-Talmudic form of Judaism. When Y chromosomes point one direction and mitochondrial DNA points another, population geneticists call it sex-biased admixture. It is not ambiguous noise. It is the forensic fingerprint of a specific class of founding event. Men arrived, carrying one lineage. Women were already there, carrying another. Their descendants built a community. And what those descendants became across 1,600 years of isolation in the Ethiopian highlands is what this episode is going to prove, and then reveal what a government did when it found out. If you've never subscribed to this channel, this is the moment, because what's coming will not leave you. Sometime between 2,000 and 3,000 years ago, a group of men crossed the Bab el-Mandeb Strait, the Gate of Tears, carrying specific Y chromosome lineages in a specific religious tradition. They founded the oldest sub-Saharan Jewish community on Earth. They left no written name, no record of departure from anywhere, no account of why they came. The science reads their chromosomes. The history reads only their silence. The Bab el-Mandeb Strait, present day, 26 kilometers at its narrowest point. The distance between the Yemeni coast and the Djibouti shoreline, divided by the island of Perim into two channels. Arab navigators named them the Gate of Grief. The currents change direction without warning. The reefs are invisible at high tide. In antiquity, this was simultaneously the most feared and most trafficked waterway between the Mediterranean world and the Indian Ocean. Sometime between the 1st and 6th centuries of the Common Era, possibly earlier, possibly in multiple waves, men carrying a specific religious tradition and specific paternal chromosomes crossed it heading southwest, toward the Ethiopian highland plateau. They did not leave a departure record. They left a haplogroup. A 2019 reassessment of Eurasian genomic signatures in Ethiopian populations traced the source of the West Asian admixture signal specifically to populations related to the ancient Levant, not to Arabia broadly, but to populations ancestrally linked to the early Neolithic farmers who had seeded Europe's agricultural revolution from the Near East thousands of years before. The signal was concentrated in the northern Ethiopian highland populations, precisely the geographic range occupied historically by the Beta Israel. The route is consistent with Aksumite trade networks connecting the Red Sea to the Levant via South Arabian ports. The men who crossed that strait in the 2nd century of the Common Era and the men who boarded those planes in 1991 share the same unbroken paternal line. The Bab el-Mandeb crossing took roughly 45 minutes by boat. The genetic signal it deposited took 1,800 years to sequence. The kingdom of Aksum was not an inland agricultural state. It was a maritime empire, dominant from the 1st through the 7th centuries of the Common Era. Its ports at Agilis on the Red Sea connected Rome, Arabia, India, and the East African interior. Jewish merchants operated in verified Aksumite trade networks, attested in Arabic, Syriac, and Greek sources. The founding of the Beta Israel required no miracle. It required a trade route, a generation of intermarriage, and local women who became mothers. The 4th century. Aksumite king Ezana converts his kingdom to Christianity, making Aksum the second documented state in history to officially adopt the faith after Armenia. Ethiopian historical records and Christian chronicles confirm that Jews in the Aksumite kingdom refused conversion and were forcibly marginalized. The Beta Israel retreated to the highlands around Lake Tana, to the Semien Mountains, where archaeologists have since identified stone synagogue structures predating any possible contact with rabbinic Judaism. What was preserved in those mountains was older than the Talmud itself, a trade route, a religious minority that refused a king's order, a mountain, and 1,600 years of genetic isolation that preserved 2 separate chromosomal inheritances, the Levantine fathers and the East African mothers, intact, unmerged, readable across 3 millennia by a machine that did not exist until 2003. For decades, the dominant academic model held that the Beta Israel were indigenous Ethiopians who converted to Judaism through cultural contact, who shared no genetic connection to the ancient Levant. The DNA partially contradicts this, but it does not fully confirm the alternative either. Both can be simultaneously true, and that is not a paradox the field has resolved. A genetics laboratory, any university campus, circa 2004, a researcher runs a haplogroup frequency analysis on a Beta Israel sample. The printout reads, 51.2% of Beta Israel maternal lineages belong to macrohaplogroup L, the deep African maternal root, the lineage tracing back over 150,000 years into the East African Rift Valley. The remaining maternal lines, 22% R0 Eurasian, 19.5% M1, a lineage that crossed the Bab el-Mandeb from Arabia into East Africa long, before Judaism existed, 5% W, 2.5% U. The researcher writes down the numbers, then faces the one question the numbers cannot answer. What does this tell us about who these people are? Every popular account of this story says the Beta Israel have mostly African DNA. This is technically accurate. It is scientifically misleading in equal measure. What they possess is the most complete layered maternal genetic record of East African Rift Valley, prehistory, and a paternal line that terminates in the ancient Levant. These are not 2 ways of saying the same thing. The 51% African maternal signal does not mean what the popular framing implies. It does not mean local conversion with no outside ancestry. It means exactly what it says. The mothers of this community, across every generation from the founding to the present, were predominantly local women. The fathers of the founding generation were not local. This is 2 separate facts about 2 separate groups of people. They have been systematically collapsed into one conclusion. That collapse is the error. The haplogroup M1 specifically traces a westward arrival into East Africa from South Asia or Arabia, a backflow that predates any Jewish presence in the region by thousands of years. Beta Israel maternal diversity is therefore a layered archive, a 150,000-year African root at the base, early Holocene Eurasian backflow on top, and then, layered over both, the specific founding event that brought Judaism. 3 separate migrations, 3 separate chapters, 1 people. The Beta Israel practice of Haimano-Judaism has no Talmud, no oral law, no rabbinic tradition developed after the 2nd century of the Common Era, but it has documented strict Sabbath observance, circumcision, Levitical purity laws, slaughter practices. When the Beta Israel were airlifted to Israel in 1991, they had never heard of Hanukkah. The holiday commemorates events from the 2nd century before the Common Era, after their ancestors had already departed. They preserved a version of Judaism that the rest of the Jewish world had moved far beyond. That preservation is not in the sequencer output. It is in the liturgy. In Ge'ez, the entire field assumed for decades that the way to verify Beta Israel Jewish ancestry was to find genomic similarity with other Jewish diaspora communities. The 2010 Nature study found no such similarity genome-wide. What it found instead is more significant: a community that preserved Jewish practice in total isolation, without the genetic integration patterns that produced every other diaspora Jewish group, which means Jewish identity, in the case of the Beta Israel, was being transmitted through something that chromosomes cannot sequence. Gamo Highlands, Southern Ethiopia, Moda Cave, 2015. An archaeological team extracts the petrous bone, the densest bone in the human skull, the one most likely to preserve ancient DNA intact. from skeletal remains. The individual was male. He died approximately 4,500 years ago in the Ethiopian highlands. He had never been within 1,500 kilometers of the Levant. He had 0 Eurasian ancestry. His genome, sequenced to 12.5 times coverage, is catalogued as MODA. He is the first ancient African genome ever successfully recovered, and he retroactively changes the meaning of every Beta Israel study published before him. Sequencing MODA's genome established a pre-admixture African baseline for East Africa, a genetic snapshot of the region before any West. Eurasian backflow arrived. For the first time, researchers could formally separate 3 distinct ancestry layers in Ethiopian genomes: the deep African Rift Valley ancestry, present before any outside contact; an early Neolithic Eurasian backflow, arriving approximately 3,000 years ago from a population related to the first farmers of Anatolia and the Levant; and the specific Levantine founding signal visible in Beta Israel paternal lineages. Without MODA, these 3 layers were inseparable. Every previous analysis of the Beta Israel had been running an uncalibrated instrument. For 30 years, researchers analyzing the Beta Israel were reading their genome against a baseline contaminated by the very migrations they were trying to measure. MODA ended that. He is a 4,500-year-old man in a cave, and he is the most important methodological advance in the history of Beta Israel genetics. The early Neolithic Eurasian backflow that MODA's genome revealed, arriving approximately 3,000 years ago from Anatolian and Levantine farming populations, is not the same signal as the Beta Israel's paternal Levantine lineage. It is an earlier, broader migration. That distinction matters enormously. It means the Beta Israel's Y chromosome signal is not background noise from the general 3,000-year backflow. It is something additional, something specific, something that arrived later through a more targeted channel. The MODA individual was found with red ochre markings on his bones, a burial practice documented across East Africa for over 100,000 years. He carried gene variants associated with high-altitude adaptation. He was completely unaware that approximately 500 years after his death, a wave of Eurasian farmers would cross the Red Sea and leave a genetic signature in his descendants that researchers 4,500 years later would struggle to separate from his own. He did not consent to being a baseline. He became one. When researchers used MODA as a baseline and ran the admixture models on Beta Israel genomic data, the Levantine paternal signal dated to a founding event consistent with the 1st through 6th centuries of the Common Era, the precise window of the Aksumite trade era. But one result produced a date that the standard model cannot account for. The signal is older than the model predicts it should be. No one has yet published the explanation. A Beta Israel village north of Lake Tana, circa the 14th century. A man observes Shabbat with his family. His wife's maternal line traces back 8,000 years into the East African Rift Valley without interruption. His own paternal line, the Y chromosome he carries, unchanged since it was first copied in the ancient Levant, has been transmitted without alteration through 60 generations of men. He does not know what a chromosome is. He knows the prayers in Ge'ez. His Y chromosome and his wife's mitochondrial DNA will both be readable by machines 700 years after his death, and they will point in opposite directions. A 2020 genome-wide study of the Bronze Age southern Levant examined DNA from 73 individuals across 5 archaeological sites. It established that the East African-related component in Arabic-speaking Middle Eastern groups is measurably higher than in non-Ethiopian Jewish groups, suggesting that the Beta Israel's paternal Levantine ancestry arrived before the large East African admixture waves that later affected Arabian populations. The signal predates the noise. If you have taken a consumer genomics test and received a Middle Eastern or Levantine percentage, you are reading the same type of signal, the same inheritance mechanism that is at the center of this legal dispute. The technology is identical. The stakes for the Beta Israel were not a curiosity. They were a verdict. The Y chromosome is transmitted exclusively from father to son, unchanged except by rare mutation across every generation. It does not blend with the mother's genetics. It does not average out across a family. It is the most direct biological thread connecting a living man to his founding ancestor. The Beta Israel's Levantine Y chromosome signal is not a historical hypothesis. It is a direct molecular inheritance. A thread with one end in a village north of Lake Tana in the 21st century, and the other end somewhere in the ancient Levant 2,000 years ago. In the 15th century, Ethiopian Christian emperors launched a sustained military campaign against the Beta Israel. The wars lasted intermittently for over 200 years. Forced conversions, executions, land confiscations. The men who survived those wars, who passed their Y chromosomes to sons who passed them to sons, carried the Levantine paternal signal through the entire destruction. The thread survived the sword. Every male line descendant of the founding Beta Israel men carries in every cell of his body a direct molecular copy of that original Levantine paternal line. If any of those men were tested in an Israeli rabbinical court between 2013 and 2019, and some were, the machine reading their sample was reading the same thread that crossed the Bab el-Mandeb 2 millennia ago. The court did not know what the machine was looking at. The machine did not know what the court would do with what it found. There is a single Y chromosome mutation, documented in the published literature, traced to a founding male ancestor of the Beta Israel, that exists in living men today who carry it without knowing what it witnessed. That mutation was present when the 15th century wars tried to end this community. It survived. The men carrying it today are in Israel. Some of them have been asked to prove they are Jewish. Gondar, Ethiopia, 1991. A 5-year-old girl named Yiddish Aina loses both parents. Her grandmother Zaituna walks with her toward Sudan, part of the Beta Israel community movement toward Operation Moses airlift points. They reach Israel. Yiddish grows up in Netanya, learns Hebrew, serves in the Israeli Defense Forces, becomes a model. In 2013, 22 years after her grandmother walked toward Sudan, Yiddish Aina is crowned Miss Israel. She is the first Ethiopian Israeli to hold the title. She later meets United States President Barack Obama at a state dinner. Her citizenship rests on a 1977 Israeli government decision that the law of return applied to the Beta Israel. That decision was made 33 years before the genome study that placed her community outside the Jewish genetic cluster. The 2010 Behar et al. Nature study was published in June 2010. The Israeli chief rabbinate's documented use of genetic testing in Jewish identity proceedings began in cases traceable to 2013. According to state attorneys' own records submitted to Israel's Supreme Court in 2019, at least 17 cases of DNA testing for Jewish status were conducted in rabbinical courts from 2013 onward, 3 years from publication to courtroom. That is how long it took for a peer-reviewed genome study to become legal evidence about a living person's identity in a democratic state. 3 years. Yiddish Aina did not choose to be a data point. She chose to compete in a pageant. Her community's genome was published without their knowledge being sought in a study conducted in Israel about people who had been Israeli citizens for 19 years before the study was submitted. The science described them. The courts reached for the science. Yiddish Aina won Miss Israel the same year the court cases began. The same data underlies both events. One is a celebration. One is a legal proceeding. The same chromosomes are present in both rooms. A community that Israeli rabbinical authorities required to undergo symbolic conversion for decades. Even after the law of return was extended to them in 1977, even after 177,000 of them became Israeli citizens, carries a genetic lineage that the dominant academic model assumed had no ancient Levantine founding component. The Y chromosome says otherwise. And in January 2020, the Israeli High Court ruled that DNA testing to prove Jewish status was permissible. The ghost did not disappear. It was given a legal address, an Israeli rabbinical courthouse, Jerusalem or Tel Aviv, 2013 through 2019. A person, a former Soviet immigrant, or a second-generation Ethiopian Israeli, presents themselves before a rabbinical judge to register a marriage. They have identity documents. They have community witnesses. The rabbinical judge reviews the documents and requests one additional form of evidence, a DNA test, not optional. If they decline, the marriage registration enters indefinite suspension. If they comply, a sample of their blood or saliva is analyzed by a genetics laboratory. The result is compared against documented Ashkenazi mitochondrial DNA markers, or in the case of a Beta Israel individual, against the genome-wide clustering patterns established in the published literature. Between 2013 and 2019, the Israeli chief rabbinate acknowledged at least 17 documented cases in which genetic testing was used to establish a family link or confirm Jewish status in rabbinical court proceedings. Rabbi Seth Farber of the Etam organization, which assists immigrants navigating Israeli religious bureaucracy, stated publicly that his organization alone represented at least 14 individuals in that period who were asked to take DNA tests. Israeli civil petitioners sought to prohibit the practice. In January 2020, the Israeli High Court rejected those petitions. It ruled that DNA testing to confirm Jewish status was permissible under Israeli law. What you just heard is not a dystopian hypothetical. It is documented Israeli legal history from between 2013 and 2020. The High Court ruling is on record. The 17 cases are on record. The item representation of 14 individuals is on record. This is not a warning about what science might become. It is a description of what it already was. The researchers who published the Behar et al. 2010 Nature study were answering a scientific question about population structure. They were not writing a brief for a rabbinical court, but the study was published in a jurisdiction where religious courts have civil authority over marriage, divorce, and burial. And the juridical use of genetic data does not require the researcher's consent. The data, once published, becomes available to any authority willing to reach for it. The reach in this case was documented. The Beta Israel's Y chromosomes carry a paternal signal from the ancient Levant. Their maternal genome is overwhelmingly East African. Both findings are real. Together, they are the forensic signature of a male-only founding migration, the specific mechanism by which Judaism entered the Ethiopian highlands 2,000 years ago. And between 2013 and 2019, Israeli rabbinical courts used genetic testing in proceedings about whether individuals were Jewish enough to marry in Israel, making the Beta Israel and former Soviet Jewish immigrants the only communities in modern democratic history whose chromosomal data was formally leveraged inside a functioning state's legal apparatus to adjudicate human identity. The sequencer became a witness. The witness was not asked to take an oath. Rabbi Seth Farber publicly called the practice scientifically flawed and Jewishly misguided. Geneticists whose research was being cited in these proceedings stated publicly that Jewish identity cannot be confirmed by genetic markers, that population genetics describes ancestry patterns, not identity categories. Their objections were documented. They were not binding. The genetic fault line the Beta Israel carry in their chromosomes, Levantine fathers, East African mothers, maps precisely onto the contemporary Israeli debate about who belongs to the Jewish people. That debate was not resolved by the 2020 High Court ruling. It was formalized. The same fault line that crosses a chromosome now crosses a legal document. Both are still open. Tel Aviv University Genetics Laboratory. A researcher opens the Behar et al. 2010 dataset on a monitor. The Beta Israel cluster is visible in the top right corner of the principal component analysis plot separated from the other Jewish groups adjacent to the Amhara and Tigrayan samples. The researcher knows this. The researcher also knows that the Beta Israel have maintained a specific, documented, pre-Talmudic form of Judaism for 1,600 years in total geographic isolation, without access to any rabbinic tradition, without knowledge of Hanukkah or Purim or the oral law, preserving a version of the faith that no other community on earth maintained. The cluster placement does not explain that. It does not speak to it at all. The 2010 Nature study itself notes, in its own language, that the Beta Israel case demonstrates that Jewish identity in this community was transmitted through mechanisms that the genome-wide clustering analysis does not capture. The cultural record of Haimano-Judaism, a pre-diaspora Torah tradition preserved for 16 centuries, is data of a different type. It is not genetic. It is not less rigorous. It is a different archive of the same event. The field has a word for what the Beta Israel preserved that the sequencer cannot read. The word is orthopraxy, correct practice, and no haplogroup frequency distribution has ever captured what 1,600 years of correct practice in an isolated highland community means as a form of transmission. The sequencer reads the code. It does not read the book the code was used to copy. Every authority that has attempted to adjudicate Beta Israel Jewish identity, from the Israeli chief rabbinate requiring symbolic conversion in the 970s to the rabbinical courts requesting DNA tests in 2013 through 2019 to the High Court ruling of 2020, has reached for a category of evidence that the Beta Israel's own tradition makes irrelevant. Haimano-Judaism does not define membership by bloodline. It defines it by practice. The legal system that has been demanding biological proof of identity from a community that has never required biological proof of identity has applied the wrong epistemology to the wrong archive. A Beta Israel Kes, a religious leader, serving a community in Gondar in the 1970s could recite the entire Torah in Ge'ez from memory, conduct all Levitical purity rituals, administer Shabbat observance, perform circumcision. He would have had no access to the Talmud, the Mishnah, or any post-2nd century rabbinic text. He was more directly continuous with the practices documented in the Pentateuch than any Ashkenazi rabbi alive. His Y chromosome would have told a sequencer that his paternal line originated in the ancient Levant. The sequencer would have been right. The court that required his community to undergo symbolic conversion was asking the wrong question of the wrong archive. There is one piece of evidence that would resolve the question of the Beta Israel's founding event with specificity. Ancient DNA from pre-Aksumite, Sites in the northern Ethiopian highlands, specifically from the Lake Tana region, dated to the 1st through 4th centuries of the Common Era. As of the current published literature, no such samples have been successfully extracted and sequenced. The founding event is visible in its chromosomal consequences. The founding event itself has never been directly read. Tel Aviv, 2024. There are 177,600 Israelis of Ethiopian descent. The Chief Rabbinate issued its formal recognition of the Beta Israel as Jewish in January 2020, 43 years after the law of return was first extended to them. Some of their children were born in Israel, have served in the Israeli army, speak only Hebrew, and have been asked by rabbinical offices to submit genetic evidence of their Jewishness in order to register a marriage. The recognition and the requirement exist simultaneously. The same person can be recognized as Jewish by one office and asked to prove it by another. This is not a historical condition. It is a current administrative reality. The Israel Democracy Institute's 2019 policy brief on genetic testing for Jewish status concluded that the practice represented the first step in the creation of a genetically based Judaism, a category that has no precedent in 3,000 years of Jewish legal tradition. Traditional Jewish law defines matrilineal descent and conversion as the two pathways to Jewish identity. Genetic testing is neither. Its introduction into rabbinical proceedings is not a recovery of ancient practice. It is a novel invention using a 21st century tool to answer a question that 3,000 years of Jewish jurisprudence never asked in biological terms. The Beta Israel did not ask to be a test case for the intersection of genomics and civil rights. They asked to come home. The science answered a question about their chromosomes. The law answered a question about their belonging. These are not the same question. They have never been the same question. If you carry East African mitochondrial lineages, macrohaplogroup L, or its derivatives, you are carrying the same maternal archive that underlies the Beta Israel's maternal genome. If you carry haplogroup J in your paternal line, you are carrying the same West Asian marker that the Beta Israel's founding men brought across the Bab el-Mandeb. These are not metaphors, they are molecular facts. The chromosomal story this episode told is not one community's story. It is the architecture of how all human communities form: men who arrived, women who were already there, children who became something neither parent had a word for. Your own genome, wherever you are watching from, is built on the same mechanism. The question of what that mechanism means, the question of what ancestry is allowed to decide about who you are, is the question no sequencer can answer and no court should try. If this episode changed how you think about identity, ancestry, and what science is allowed to decide, subscribe and share it with someone who needs to think about this too. The next episode goes somewhere you will not see coming. The Beta Israel crossed the Gate of Tears once, 2,000 years ago, in a direction the history books did not record. They crossed it again in 1991 in 747s, over a sea their ancestors crossed by boat. Their chromosomes carry both crossings, and in 2020, a court decided that reading those chromosomes was legal. The court has not yet decided what the reading means. Neither has the science. Neither have we.